Okay, hello everybody. Welcome to NLML seminar. Today I'm pleased to present Jim Cloern, who is a senior scientist emeritus at the USGS in Menlo Park. His research over 4 decades has focused on San Francisco Bay and addressing comparative biology and biogeochemistry of estuaries to understand how they respond as ecosystems to climatic and hydrologic variability, as well as human disturbance. So his investigations over the years have included studies of primary production, nutrient cycling, ecosystem metabolism, invasive species, as well as past and projected future response to a changing climate. Before we start the seminar, I'll just go over the Zoom etiquette. All audience participants will be muted throughout the seminar. Please do not attempt to turn on your video or share your screen during the talk. And there will be an opportunity for the audience to ask questions at the end of the talk, just like a normal seminar. Once the speaker has finished his talk, you can use Zoom's raise hand feature to notify me that you'd like to ask a question. And the raise hand feature is located just under the Participants tab at the top of the Zoom window. So thank you, and I'll let Jim present his talk now.
Well, first of all, I want to thank you for this invitation. I'm sitting up here in the Bay Area on a rainy Thursday afternoon, wishing I was in Moss Landing with you all. I know you're all anxious to be back in the seminar room, and it is going to happen. I'm going to play a video of a seminar that I gave at the Institute of Marine Sciences at the University of North Carolina. And the title, I guess the title of this on this slide is different than the title I indicated, but the topic is the same. I'm going to talk about in this presentation about patterns and processes of change where rivers meet oceans, and in particular from our long-term study of San Francisco Bay. So let me play this video. I'm going to tell you that at the very end of the video, I'm going to make reference to being in Morehead City in North Carolina. So just at the end of the talk, imagine that you're sitting on the shores of the estuary of North Carolina, the Neuse River estuary. So let me fire this up, and then I'll be happy to answer any questions you have. This is about a 40-minute presentation.
Talk about estuaries, and you all know that estuaries are places where fresh water from rivers meets and mixes with ocean seawater. And this image here of San Francisco Bay, both on the screen and behind me, San Francisco Bay is an estuary. It's an estuary that I spent a career working on. I joined a USGS research team in 1976, and that team actually started in 1969. So the very first USGS sampling cruise in San Francisco Bay was in the spring of 1969. I'm going to start this talk with a short film clip to give you a sense of what estuarine science was like in that era when this program began. And in the late 60s and 70s, there really wasn't a bona fide science of estuarine oceanography or ecology. We're kind of feeling our way through this. And of course, in the decades since, we've learned a lot. So let me start with this film clip to give you a sense of the historical way in which we were studying estuaries, how this USGS program launched, and then we will go from there. And I will let you know in advance, this is a really coarse video. This was well before we had high definition cameras.
What's going on inside the Golden Gate? It's a question to dream about before the day's research begins.
We are doing
the group together?
Well, I think we are together as a group because primarily we're fascinated by the estuary, and we're fascinated for several reasons. One of which is that we know the scientific community knows so little about estuaries.
Now they're finding out. It's basic research. The Geological Survey, fortunately, has no vested interest in the result.
Our agency doesn't have a particular regulatory function. It doesn't have a particular mission of dredging harbors or building dams, selling water. We were just set out there and they told us, see how San Francisco Bay works.
To the west coast, the bay is fed by the two great rivers of the California Central Valley. That makes it not just a saltwater bay, but a huge estuary, meeting place of fresh and salt water. Seeing how San Francisco Bay works means, first of all, understanding the delicate balance between river and ocean. These basic properties affect everything else. The research group's water chemistry expert on board the Polaris is Dave Peterson.
Now we're in the no zone area, which is in San Pablo Bay, and we're going to check how much fresh water we have at the surface based on its salinity and how much salt water we have at depth by measuring the salinity. So first we'll start out with a probe at the surface. Lower the probe to the surface.
Peterson's been developing this water analysis system for the last 5 years. The probe continuously pumps water samples back to an instrument cabin. There, many different dissolved chemicals are automatically measured and recorded.
Okay, the salinity of the surface water is 7.2 parts per thousand. And to give you an idea, normal seawater is about 33, 34 parts per thousand. Now I'll put it down to the near bottom water, which is 8 meters deep. Lower the probe to 8 meters. Now you can see that the salinity is increasing. The temperature is starting to drop and the depth gauge moving down. The salt content is 13 parts per thousand. That's roughly a doubling increase in the amount of sea salt at depth. So that's a look back in time, and obviously a lot of things have changed since the 1970s. I imagine it caught your attention that everybody in that scene was a guy, and if you went on one of our sampling cruises today, the scientific crew is right now all female. So a lot of things have changed since the 1970s, and I'm going to focus on how this estuary changed. And I think that maybe the most important general lesson that's emerged from this program is the surprising fast pace at which the estuary changes. And I'm going to give 6 examples of change that's been captured by observations made over periods of decades. I want to talk about what we've learned since that era in the early 1970s. And I want to start with this physical map of California to set the stage. So you know that California has 2 mountain ranges, the Coast Range and then the Sierra Nevada Range. And the area from the western slope of the Sierras to the Coast Range is this large watershed. So that's the runoff in this watershed is the source of fresh water that makes San Francisco Bay an estuary. That runoff is coalesced into the Sacramento River, which flows from the north. The San Joaquin River flows from the south. They converge in this area that we refer to as the Delta. And so with this kind of setting in mind, if we zoom in to look closely at the estuary, this is the region where the Sacramento River and the San Joaquin River converge. So this is the upper part of the estuary is low salinity. There's a salinity gradient from freshwater to seawater, near seawater at the Golden Gate. And then we also have this area to the south, which is a marine lagoon type estuary. And the examples of change that I want to show you are examples of change that originated far upstream and were carried by river flow, or in this densely populated urban landscape, or something that changes that took place in the Pacific Ocean. So these boundaries with oceans, urban settings, and rivers are characteristic features of estuaries that drive their variability. So the first change that I want to talk about occurred in the late 1800s. It's a change that occurred hundreds of miles away from San Francisco Bay on the western slope of the Sierra Nevadas, but had a big downstream effect. And that was a human activity. This is an old photograph from the 1800s, and I hope you can see that these streams of water from pressurized with these jets that were tearing down hill slopes so that they could extract ore from which they then extracted gold. This was the end of the kind of the last phase of the gold rush era. And as you can imagine, this was a major physical disturbance in the upper watershed. It had a big effect downstream on the rivers in the bay. A USGS scientist in the early 1900s did a calculation, and he estimated that about 1 billion with a B cubic meters of sediment were deposited downstream in San Francisco Bay. And of course, this had a big effect on the bathymetry of the bay. And one of the ways that we know that is by comparing different bathymetric charts over different historical periods. This was also done by a colleague of mine at the USGS, Bruce Jaffe. So to get you situated, this is a particular embayment in the northern part of San Francisco Bay. And the map here, the color map here, shows differences in water elevation comparing the very first bathymetric survey done in 1856 with the next one in 1887. So over this 30-year period, Bruce calculated that just this region alone accumulated about 250 million cubic meters of sediment. And the accumulation is shown in red here. So the red areas are areas that became shallower as a result of sediment inflow inputs to the estuary as a consequence of hydraulic mining. And the mudflats expanded by about threefold, and the channels shallowed by up to 12 feet. So here's an example of change, a pretty abrupt change in the morphometry of the bay as a result of human activity far away from the estuary. The second example of change that I want to give follows that, and it began in the early 1900s. It's another example of human modification of the watershed that had downstream effects. And that modification was a result of the fact that in this era, beginning in around 1900, we began to dam all of the rivers that converge and are the tributary rivers to San Francisco Bay. And this graph here, this black line, represents the growing cumulative storage capacity of reservoirs as they went online. So the large one in 1945, Shasta Dam, went online, and you can see this step increase in the cumulative storage capacity. So the dam building era ended around 1980. All of the large rivers are dammed. And of course, one of the main downstream consequences of damming rivers is that sediments from erosion of the land surface that naturally would flow from the soils into the rivers and then down into San Francisco Bay, much of that sediment is now retained behind dams. And so we have reduced the input of sediments from the watershed into the estuary. This is a common consequence of river damming. And so we have observations over, in this case, a 35-year period, showing changes, decreases in, this is a log scale of suspended particulate matter. So this is basically suspended sediment concentrations at one location in the estuary. So here's a big change in the estuary, again, resulting from changes that took place in the watershed. And there are, of course, consequences of decreasing sediment input to estuaries. I'm going to show 3 here. First of all, comparing more recent bathymetric surveys, Bruce Jaffe showed that mudflats are now being lost from this region of the estuary, and that's important for many reasons, including the fact that San Francisco Bay is on the Pacific Flyway, and waterbirds use these mudflats as feeding habitat. Also, the decrease of sediment supply and concentration has increased the transparency of water, and this has had biological consequences, including allowing proliferation of introduced invasive aquatic plants, rooted plants like the Brazilian water reed, Agaricus densa, which has become a nuisance problem. And the third implication or consequence of reduced sediment supply is there's growing uncertainty now about the sustainability of tidal marshes when we consider the fact that the sediment supply required to sustain marshes is decreasing, coupled with rising sea level. Okay, the third change that I want to talk about also is something that happened inland, away from the estuary. And this was the management of water, both by consuming fresh water in the upper part of the watershed and exporting water into the state and federal water projects into these large canals. So the result of both upstream consumption and exports has been an alteration in the quantity and seasonal timing of freshwater inflow to the estuary. So this graph here shows annual values of the ratio of the volume of water that was exported divided by the quantity of fresh water that was inflowing in towards the estuary. And you can see that big changes happened from the 1950s to the 1980s, when this ratio now is reaching a median value of about 30%. You can see that there's a lot of variability from year to year. So consequences of water management have resulted in an almost 40% reduction of freshwater inflow to the estuary. And estuaries are where river water meets seawater, and if we're altering the quantity and timing of freshwater, we're altering the characteristics of the estuary. So one of the outcomes has been, as we are allowing less fresh water to flow into the estuary, it's being used downstream for municipal water supplies and irrigated agriculture. One of the consequences has been an intrusion of salinity as river flow has decreased. So this is a human-induced migration of the salt field from the salty part of the estuary into the low salinity part of the estuary. And we estimate that during the autumn season, when this effect is largest, there has been an 8-kilometer intrusion of salinity as a result of our water management activities. And of course, changing the salinity distribution has many biological effects, including effects on the quality of habitat for different species that live along different regions of the salinity gradient. This is a graph showing another kind of change. This is change dating back to the late 1960s to the 21st century. The dots represent an index of habitat quality for one of the indigenous species that lives in the low salinity part of the estuary. This is the Delta smelt. And this habitat quality index has 3 components: a component of water temperature, water transparency, and salinity. And so as a consequence of decreasing sediment supplies, increasing water transparency, and increasing salinity, the habitat quality for this indigenous species of fish has decreased over these decades. So these changes in the salinity field and in the transparency of water as a result of human activities have an effect on habitat quality for the species. And not surprisingly, the species that evolved in the Delta, the indigenous species, including the Delta smelt and the longfin smelt, their populations have declined significantly over the last 5 decades or so. These are log scale plots of abundance indices of the Delta smelt going back to the late 1960s and longfin smelt. And you can see orders of magnitude decreases in abundance. And this change in habitat quality, this reduction in habitat quality, is one of the contributing factors to these to these population declines. These are both species that are protected under state and federal conservation laws. Okay, so let's move from the upper part of the estuary to what we refer to as the South Bay. And this is, again, a marine lagoon estuary. It has less direct freshwater inflow, but it has this direct connection to the Pacific Ocean through the Golden Gate. I want to show changes that took place in this lower sub-embayment in the 1970s. And you can see this densely populated urban landscape around this marine lagoon. There are now 7.5 million people that live around the bay. We discharge our waste, treated waste, through 42 sewage treatment plants that discharge into the estuary. One of the largest is this one here in the lower right. This is treats the San Jose region in the southern part of the Bay Area. And the change that I want to talk about in this small region with relatively restricted circulation took place in the 1970s. And I'm starting with data that were collected before our program began in the late 1950s and early 1960s. And these dots represent individual measurements of dissolved oxygen concentration in units of milligrams per liter. And I've highlighted the value 5 here because 5 milligrams per liter is a state metric defining whether water quality is meeting the requirements for fish species sensitive to low oxygen. And you can see that in that era, in the late 50s and early 60s, it was common, these red dots, for oxygen concentrations to fall below 5 milligrams per liter. And in fact, during the summer of those years, the water in that region of the estuary was anoxic. And these episodes and seasons of hypoxia or anoxia were occurring regularly in US rivers and estuaries and lakes, and were a primary motivation for passage in 1972 of the Federal Clean Water Act. And among other things, the Clean Water Act mandated that we increase the treatment of sewage discharged into estuaries to reduce the impacts of poorly treated wastewater on oxygen concentrations in rivers and estuaries. And the law targeted specific treatment processes to remove organic matter from wastewater effluent that we refer to as BOD, biological oxygen demand. It's essentially a measure of organic matter that's available to be metabolized by microbes, and then the reduced form of inorganic nitrogen, ammonia. So the reason for the low oxygen concentration was these very high annual loadings of BOD and ammonium in the 1950s, 60s, and 70s. And the San Jose sewage treatment plant implemented advanced treatment to greatly reduce BOD and ammonium. That was implemented in 1979. And you can see this abrupt step decrease in annual loadings of BOD and ammonia. So we know how to treat sewage, we know how to remove organic matter and reduce forms of nitrogen. And this graph shows the consequences for water quality changes in the estuary, in that small embayment to the extreme south. So to the left here, we see these data of these many red dots showing episodes of hypoxia or anoxia, this transitional period in the 1970s. After 1979, basically, these occurrences of hypoxia, anoxia have disappeared. So we rarely see oxygen concentrations below 5 milligrams per liter. And nowhere in San Francisco Bay now do we have problems of low oxygen concentrations. So this problem was solved in San Francisco Bay, and of course it was solved in many other rivers and estuaries around the US. We think, for example, this is a really good example, the Potomac River. So now let's move back to the salty upper part of San Francisco Bay, and I'm going to talk about a very different kind of change that developed in 1986. These changes were brought about by a discrete event, and that event was the introduction of a non-native clam. This is a small clam, Corbicular amurensis. It's indigenous to rivers of Asia. And it was first discovered in this upper part of the estuary in the autumn of 1986. And within a year, by the end of 1987, its population had exploded in the low salinity parts of the estuary. It carpeted the bottom sediments. And the bar graph here shows the annual abundances in numbers per square meter at a particular location in the low salinity part of the estuary. And this organism, this invasive species, succeeded because it was introduced to a vacant niche. And the niche is a region of the estuary that becomes fully fresh water during the high flow season. So the local marine bivalves don't have the osmotic capacity to survive for months in fresh water. This particular species does. It has an exceptional capacity to survive in fresh water. It can't complete its life cycle, but it can survive. And it can thrive in salinities that are brackish. So it was introduced almost certainly by discharge of ship ballast. It was discovered probably immediately after it was introduced. Its population exploded, and it generated a remarkable restructuring of the plankton community in the low salinity parts of the estuary, not surprisingly considering these abundances. So let me walk you through the changes that took place in the planktonic food web of this region of the estuary after corbicular was introduced. So again, at the top here is this bar graph showing the annual abundances of corbicular. And then below that are annual indices of phytoplankton biomass, that's chlorophyll, and then abundances of three important components of the zooplankton community: the rotifer synchaeta, the calanoid copepod eurythemera, and the mysid shrimp Eomysis. And these four bar graphs at the bottom are organized so that they're centered around the long-term mean. So bars above the long-term mean are blue, and bars below the long-term mean are red. And you can see a discrete downshift in phytoplankton biomass. Phytoplankton primary production after this introduction was reduced by a factor of 5 because of this strong top-down control by the filter feeding introduced clam. The phytoplankton food resource was greatly reduced, and as a consequence, losses in the abundance of rotifers, calanoid copepods, and mysis. And in fact, this calanoid copepod eurythemera and Neomysis are basically absent from the low salinity parts of the estuary now. So a remarkable transformation of the pelagic food web following the introduction of this invasive species of clam. And those of you who know similar stories in the Great Lakes and in the Hudson River know that this isn't a unique story to San Francisco Bay, this particular invasive species is, but similar kinds of changes took place in the Great Lakes and the Hudson River after introductions of the non-native zebra clam, zebra mussel, and quagga mussel. So really abrupt, persistent changes in biological communities as a result of one species introduction through discharge of ship ballast. And those components of the zooplankton, in particular the rotifers and calanoid copepods, and including their nauplii, are key food resources for these small planktivorous fish that we're trying to protect, trying to sustain these populations. So in addition to changes in habitat quality, there also have been these large changes in the food supply to these planktivorous fish. And these changes also are contributing factors to the really dramatic population declines of these species that we're trying to protect. Okay, so the last change that I want to talk about is another discrete change. This one took place in 1999. And all of the changes that I've talked about so far are the result of human activities either far away in the watershed or introductions of species into the estuary. The change that I want to talk about next is a change that didn't take place in the estuary, but rather this was a large-scale change that took place across the entire North Pacific Ocean. I think some of you probably know this story. So I want to start with this figure from a paper published by Emmanuel Di Lorenzo. And you all know that over the last couple of decades, we have realized that there are two important patterns of atmospheric forcing of the North Pacific Ocean. One pattern is called the PDO, the Pacific Decadal Oscillation, and the other is the North Pacific Gyre Oscillation, the MPGO. And these color contours here represent distributions of sea level pressure. And you can see two very different patterns. The PDO pattern is one gyre system. The MPGO is a pair of counter-rotating gyres. The arrows here represent wind vectors. And I'm not sure how easy it is for you to see this, but during the PDO pattern, the alongshore winds of the coast off of San Francisco Bay are relatively weak, whereas in the MPGO forcing, maybe you can see these wind vectors that inside this blue ellipse here are relatively strong and flowing from north, carrying flows from north to south. And the north to south alongshore winds are important because those are the winds that drive coastal upwelling, that brings deep nutrient-rich water to the surface. So the PDO forcing pattern is one of relatively weak upwelling, a short upwelling season, and the MPGO pattern is one of relatively strong upwelling and a longer upwelling season. So as a consequence of these physical forcings, the PDO is a pattern of relatively low primary productivity in the coastal ocean adjacent to San Francisco Bay. The MPGO forcing is a pattern is one of relatively high primary production in phytoplankton biomass. So what's intriguing about this from the perspective of San Francisco Bay is that during the first few decades of our program, the North Pacific Ocean was in the PDO pattern. And then in 1999, it abruptly shifted to the MPGO pattern. And this functioned in a way like a natural experiment because we had a couple of decades of observations in the bay. And then all of a sudden, a flip to this other phase of the North Pacific Ocean that gave us an opportunity to see if there were changes that would take place inside the estuary, in particular regions of the estuary closely connected to the coastal Pacific through tidal exchange, for example, in organism migrations. And what we observed caught us completely off guard. The changes that we found after this climate shift took us several years to understand, and I'm not sure that we fully understand them. But the changes that took place within a year or two or three after 1999 were record high abundances of different species of fish, crabs, and shrimp inside the estuary, in the salty part in this marine lagoonal South Bay. And at the top here, I'm showing English sole, but there were 5 different species of demersal fish, including English sole, that had record 1999, 3 different species of crabs, including Dungeness crab, 2 different species of shrimp, and from our program, phytoplankton biomass increased. So this is chlorophyll. So these bars here are structured the same way that I show these biological changes in the upper estuary. They're centered around long-term means. Red bars represent abundances or biomass lower than the long-term mean, blue bars above. And you can see for this assemblage of demersal fish, crabs, or shrimp, within a year or 2 or 3 after abrupt increases to record high numbers, and again, increasing phytoplankton biomass. So this is a remarkable change in biological communities that was synchronous with a shift in the atmospheric forcing of the North Pacific Ocean, the shift from the PDO to the MPGO. And we don't fully understand the link between that climate shift and this restructuring of biological communities. But the idea that we have as an hypothesis is that the shift from the PDO to the MPGO initiated a trophic cascade. And the of bottom-feeding predators, demersal predators, including bottom-feeding fish, crabs, and shrimp. They prey on invertebrates that live in and on the sediments, including bivalve filter feeders, the herbivores. And in the salty part of the estuary, bivalve filter feeder abundance was high, and they played an important role in regulating phytoplankton biomass. So as the predator and their predation pressure increased, the abundance of herbivores dropped substantially. That released the grazing pressure on phytoplankton and allowed the primary producer biomass to increase. This is our about the linkage between this large-scale climate forcing and biological communities inside the estuary. Now, the key here is that most of these species, the fish and crabs, are marine species. They reproduce in the coastal ocean, and their juveniles settle to the bottom and then migrate into the estuaries where they feed, grow, and spend the first year or two of their life history. So these increased abundances of flatfish and crabs were increased abundances of these juvenile forms that can prey on bivalves and other invertebrates. So we've known for a long time that these multi-decadal climate drive variability, large amplitude variability in the abundances of different fish species. We think about sardines and anchovies as an example, a well-known example. But I think this might be the first example of how a shift in one of these large-scale oceanic forcings has big biological effects that propagate into an estuary. Okay, so looking back at my colleague Dave Peterson in 1973, to wrap this up, what are some of the general lessons that we've learned from 5 decades of research in San Francisco Bay? And I'll give 3. The first is that estuary and many other estuaries in being something like an equilibrium state. When I was a graduate student taking courses in ecology, we talked about equilibrium and oscillations around equilibrium states, but everything that we've looked at, biological communities, habitat quality, morphometry of the bay, sediment inputs and turbidity, freshwater inflows, timing and quantity, every one of those components that we look at has changed. Virtually everything that we measure now in 2020 in the bay looks very different from what it looked like in the 70s. So these are systems that appear to be in continuing states of change, and we think a lot about how these changes might unfold as other forcings of change, like warming and sea level rise and increased human populations in the watershed unfold in the future. The second is, I think that this lesson has importance for developing effective policies for protecting water quality, protecting habitat quality, restoring, sustaining biological diversity in estuaries. So lesson number 1 from my perspective is that effective perspective that recognizes estuaries are driven by all kinds of forcings. The amount and way in which we treat sewage, other contaminants, the way in which we manage fresh water, effects of introduced species, the way we manage fresh water and its effect on sediment supply, and these large-scale climatic forcings. And I haven't even talked about other things like warming, sea level rise, landscape change, pollutants, nutrient enrichment. Our policies seem to be focused on regulating individual stressors, but I think it's important to understand our change in estuaries is driven by many different forcings, and we have to take into consideration the effects of all of those forcings. The second lesson is that effective policies are going to be adaptable, and that means they're going to be visionary. They're going to be built around visions of change, anticipating the kinds of changes and how policies would react to those changes. And one of the reasons why policies need to be adaptable is that surprises happen. This response of the Bay communities to the shift from the PDO to the MPGO was completely unexpected, completely caught us off guard. And it's example, because of these surprises, why we need to have policies that are adaptive. And then thirdly, policies really have to take a long view. This is a time series going back to 1900 of the PDO. And you see these multiple decadal periods when the PDO is in its negative phase and then oscillates back to its positive phase. And I've shown you that the bay is a very different system when the PDO is in its negative or positive phase. So policies need to take into account the effects of these multi-decadal oscillations and maybe even have different policies depending on whether we're in the positive or negative phase of the PDO. And we certainly don't know how this climate pattern is going to change in the future. And then lastly, virtually everything that I've shown you are data from monitoring programs that have been sustained over periods of decades, not just the USGS program, but other programs as well. And it's difficult for me to imagine developing policies in the absence of information and discoveries that come from monitoring. So we need monitoring to detect change. We need monitoring to explain the underlying process of change. We need monitoring to inform policies. Clean Water Act is one example. And we need monitoring to measure the outcomes of those policies. And last thing I want to say, shout out to my buddy, Hans Perl. The monitoring imperative is not unique to San Francisco Bay, it's universal. Hans has launched this wonderful program on your estuary. I know from discussions with Hans and from my own experience how difficult it is to keep these programs going. And I wish Hans the best, and I know he wishes me the best in keeping these programs going. So I will end this now. Thank you very much. And I'll try to see if I can answer your questions.
Oh, thank you very much. It's a fascinating look at multiple decades worth of data of a complex system. So now we'll open it up to questions. And just a reminder of how this works. You can use the raise hand feature to let me know that you'd like to ask a question. At one point, this was under the participants tab, but I know that I can find it now under the reactions button on the bottom panel of Zoom. And if you can't find it, then feel free to let me know in the chat if you have a question. And I have a question while we're waiting for people to virtually raise their hands. You mentioned at the end that it's really impossible to predict what will happen with climate change, but do you have any clues in recent data, like from the North Pacific heat wave and subsequent El Nino event? Were there any shifts that you could notice in that time, or is it just not long enough to really say anything definitive?
So of course, we're interested both in trends of change over time, both those that have taken place in the past. Can you hear me okay? Does it sound okay? Yeah, but also, how are changes going to unfold in the future? And I think the regulatory agencies in California, like the California Department of Water Resources, the state water quality control boards, the California Department of Fish and Wildlife, all of them have scientific staff who are spending good chunks of their career now trying to anticipate, imagine, project how changes in water quality, water management, biological communities will unfold over the next century of continued global change. So in California regulatory agencies, the challenge of anticipating climate change driven changes, not only in the estuary, but in the watershed, in the coastal ocean, and in the urban setting as well. So California is taking this seriously. And as you know, there are many different manifestations of climate change that we need to be thinking about, like sea level rise and our adaptation to sea level rise. So just as one example, one plausible scenario in the urban landscape is that a policy response to to continued sea level rise is going to be hardening of shorelines to protect the urban centers that surround the bay, and there are going to be consequences of hardening that shoreline, including changes in upstream propagation of the tides. And so an upstream response to hardening shoreline around the bay is going to be a change in what we call the tidal excursion and the upstream transport of salt. So an unanticipated outcome, at least one that we hadn't thought about a couple of decades ago, of our responses, our policy responses to rising sea level rise, is going to be a change in the salinity distribution of the estuary. Another big concern related to sea level rise is we have spent hundreds of millions of dollars, probably now over $1 billion, in purchasing salt ponds around the bay that historically were salt marsh. And the idea was to purchase those areas, breach levees, allow tides to propagate into these salt ponds, and restore tidal marshes. And I think that on the order of 15,000 acres of tidal marshes either have been restored in the last couple of decades or are being planned to be restored. It's one of the largest areas of wetland restoration in the western part of the United States around San Francisco Bay, and we've been successful. We have seen salt marshes and their biological communities return over the last couple of decades. But the combination of sea level rise plus reduced sediment inputs to the estuary that I talked about, partly as a result of damming rivers upstream, means that the long-term sustainability of those new tidal marshes, restored tidal marshes, is really unknown. So there's an example of interactions between two different kinds of human activities: damming rivers that reduce sediment supply that's required to sustain marshes, and then coupled with elevational changes in water levels in the bay as a response to sea level rise. So this is the challenge, the challenge that we've been so successful at restoring salt marshes, but there's long-term sustainability is unknown because of these multiple interacting factors like human-driven changes in sediment supply, as well as water elevation in the estuary. That's a long-winded way of saying we're thinking hard about this, and we're putting a lot of effort into anticipating what kinds of changes will unfold and how would we adapt to those changes in the future.
Okay, thank you very much. And we do have some questions now. So I see that Christine Wasson has a question, and I'll ask her to unmute right now.
Kirsten, hi.
Hi, I don't seem to be able to turn on my video, so you can't see me because the host has stopped it. But hello, I'm a big fan, of course, Jim, of your work, and what we've done in Elkhorn Slough has been very inspired by your work with eutrophication and climate forcing and so on. But my question was about the decreased sediment supply. You showed that graph of the dams going in, the sort of step function, but it looked like not a whole lot had happened after 1980 on that graph. And yet the decreasing sediment supply graph you showed, was post 1980. It's an issue we face too, this really low sediment supply. What do you think is behind it for the post 1980 decrease?
So everything is complicated, right? Sediment supply is determined by more than just holding sediments behind dams and rivers. So a colleague of mine, geologist Dave Scholhammer, has been working on that problem for decades. He spent a good chunk of his career at the USGS working on sediment supplies and geomorphology of the bay. And he wrote a really nice paper that answers that question. And his interpretation of what's happened is we need to think about changes over periods of multiple decades. So we had 30 or 40 years of hydraulic gold mining where we were greatly increasing, we were mobilizing sediments in the watershed, and we were greatly increasing the supply of sediments to the bay and deposition of those sediments in the bay, as well as in the rivers that are flowing into the bay. Once we stopped that, we reduced that man-made increase in supply, but that mass of sediments remained in the rivers and in the bay for many decades. And gradually, primarily as a result of big storms and floods, that pool of sediments that was deposited in the rivers and in the bay as a result of hydraulic gold mining has been transported out of the bay into the coastal ocean. So two things are happening. We've withheld sediments behind dams, and then that big pool that was available to be carried from the rivers, these legacy sediments into the bay, has basically been washed out now. And the big washout occurred during the major floods of 1998 and 1999. And so there are a couple of things going on: reduced sediment supply, and then gradual, over a period of decades, mobilization of that large mass of sediments that was deposited in the late 1800s. So multiple things determine sediment supply to the bay, and now we've just reached the point where that pool is gone. And that's the explanation for why sediment concentrations in and supply to the bay have continued to reduce even after the dam building era ended in around 1980. Does that make sense? Does that? Now, about Elkhorn Slough, what the effects of that would be on Elkhorn Slough, I don't know. You're muted, Krista.
I'm trying to allow, okay.
Oh, okay. I couldn't unmute myself again. Yes, thank you. That's helpful. I think it's river diversion and lack of flood events for us at Elkhorn too, but
Okay.
I should
Yeah.
turn it over to somebody else to ask a question. But, greetings from the sister estuary to the south.
Well, one of these days we should write a paper about the similarities and differences in these two estuaries that aren't that far apart on the Pacific coast.
Yeah. That'd be great, and trends in eutrophication and so on. We'd have to focus on the salty side since we don't have a freshwater side to ours. But
yep.
All right, thank you.
Thank you, and we have another question from Amanda Kahn.
Hi, Jim. Thank you. That was a really interesting synthesis of, yeah, a lot of work. Oops, trying to lower my hand. There we go. I study suspension feeders, sponges, a lot. I actually remember a paper of yours from, I think, the 1980s on suspension feeders, and so my eye was caught by the corbicular graph, and I'm curious if anyone has looked at the effect of corbiculas feeding on the water around them, and if they are affecting water quality or clarity or nutrient fluxes through them beyond the community level.
Are you talking about in the salty South Bay part of the system or in the upper low salinity part of the system?
Well, where they invaded, the kind of the plot you had where their abundance is really increased. That was the South, right?
No, where their abundance is increased, that was in the low salinity part of the estuary because there weren't any marine bivalves there. So it was a vacant niche that it occupied, but it's distributed along the whole salinity gradient. The species is present along the entire salinity gradient, and it's present in the South Bay. It's present in the salty parts of the bay. It didn't have as big of an effect because when it was introduced, there was a well-established community of marine bivalves in the South Bay. What really set it apart in the North Bay was that it was a vacant niche. There just were not bivalve filter feeders there, and so it had this really big impact. I'm trying to think, well, there are a couple of other responses to that invasion that people have looked at, and some have been surprising. So Wim Kimmerer works on the zooplankton communities in the bay, and he and students did a really nice study showing that not only can corbula feed on phytoplankton, but also on zooplankton, nauplii of copepods, for example. And so not only are they outcompeting copepods for the food resource, they're acting as a predator as well. They're preying on the nauplier stages. In terms of geochemistry or biogeochemistry, not much has been done. I had a postdoc who was really interested in the effects of bivalves on carbonate chemistry in estuaries. And he did a really interesting calculation of the amount of CO2 that was produced by the calcification process of corbula in the upper estuary. And it had a big effect on alkalinity the water. In terms of nutrient cycling, I don't think anybody's done anything with that. There's so many questions that could be addressed about the effects of this non-native species, because it was such a profound change in the biological communities. And in a community where bivalve and other filter feeders in the benthos just weren't there, to all of a sudden they carpeted the sediments, thousands of individuals per square meter. So we know a little bit about the biogeochemical responses, but not much. There's a lot left to learn.
Right. Oh, very neat. Well, thank you very much.
Okay.
Okay, and I did get one more question in the chat, and this is from Scott Schaefer, and it sounds like his sound isn't quite working, so I'll ask it for him. He asked, what changes have occurred with regard to contaminants in the bay?
Okay, yeah, there is a lot that has been done on contaminants in the bay. The early studies in the early 1970s were led by Sam Luoma, who was at the USGS for a good chunk of his career. And he did work primarily on trace elements, nickel, cadmium, silver, copper, chromium. And when he started taking measurements of dissolved trace element concentrations in the bay, in sediments, and in biota, in the 1970s, the South San Francisco Bay was one of the most polluted estuaries in the world with respect to trace elements. For example, the silver concentrations in the sludge, the sewage sludge from the city of Palo Alto, was high enough that people were paying for that sludge to extract the silver. That was largely the result of a photographic processing plant in Palo Alto. So this is when we were using silver to produce prints. And we have made really amazing advances in reducing trace element concentrations in the bay, not only by improving wastewater treatment processes, but also controlling inputs to wastewater treatment plants from the electronics industry. So it really is a remarkable success story, the extent to which we have reduced toxic trace element concentrations in the bay. In the 1970s, the copper concentrations and silver concentrations were high enough in the bay that native species of clams were having a hard time reproducing because the waters were so toxic. And that degree of impact has completely disappeared. So that's the good news story. There are a couple of bad news stories. One is legacy contaminants, things like PCBs and DDT that were banned in the 1970s or earlier, still have very high concentrations in the sediments. Their lifespans are long, and they're going to be there for many decades to come. And the other is the challenge of emerging contaminants, things like pharmaceuticals. And now there has been disturbing, but also really important work done by the San Francisco Estuary Institute to measure concentrations in forms of microplastics in the bay. And microplastic concentrations in water and in consumers like filter feeding fish and copepods are surprisingly high in San Francisco Bay, higher than many other places that have been sampled. So the contaminant stories are varied. There are good news stories. There are stories that are going to require patience with these legacy contaminants. And there are alarming stories about emerging contaminants like pharmaceuticals and this what appears to be an important widespread problem of microplastics. So good news stories and bad news stories with respect to contaminants.
Well, thank you. It'll certainly be interesting to see what the future holds, and hopefully there's some good news in store.
Well, we need to keep the sampling going. We just need to keep these observational programs going.
Definitely.
Okay, thank you. Thank you very much.
I don't see any other questions, so let's thank Dr. Cloern and give him a virtual hand. I really appreciate you coming to talk to us.
Thanks for your patience, listening. Thanks, Tom.
Thank you.
Bye-bye.